Histology of trachea. Trachea is a conducting airway. It's a firm tube which conducts air and also clears the air of suspended particles. The trachea wall is made up of 4 layers: mucosa, submucosa, cartilage, smooth muscle layer, and adventitia. The mucosa includes the epithelium and the lamina propria. The epithelium is pseudo-stratified ciliated columnar epithelium, which is important for removing the foreign particles. The firmness of the trachea is because of the hyaline cartilage rings, which are C-shaped. So these two important points are the identifying points for the trachea. These are very important for the function of the trachea. The lamina propria is the connective tissue component of the mucosa. The submucosa is a dense connective tissue, and the adventitia is the fibroelastic connective tissue. Now we will see each layer in detail. The epithelium of the trachea is the pseudo-stratified ciliated columnar epithelium, which is also called as the respiratory epithelium, because the major part of the conducting airway is lined by this epithelium, and this is the major epithelium of the trachea and the bronchi, so it is also called as the tracheobronchial epithelium. This epithelium has various cell types of different sizes. So the nuclei, as you can see here, the nuclei are present at various levels, giving a stratified appearance, as though the cells are present in different layers. So it is called as a pseudo-stratified. Why pseudo? Because all these cells, they lie in a single basement membrane. There is a single layer of cell. It appears as though there are many layers because of the presence of nuclei at various levels, but actually all these cells lie in a single layer. All these cells rest on a basement membrane. So it is called a pseudo-stratified ciliated columnar epithelium. The predominant cell type is ciliated columnar cell. So it is called a pseudo-stratified ciliated columnar epithelium. The various cells of the respiratory epithelium are ciliated columnar cells, mucus cells or the goblet cells, basal stem cells, and the brush cells, Kolchitsky cells or small granule cells, Clara cells, which are seen in the bronchiolar level. The predominant cell type is the ciliated columnar cell. The other important cells are mucus cells and the stem cells. These cells, the brush cells, the granule cells, are present in smaller quantities. All these cells rest on a thick basement membrane, which appears as a homogeneous pale staining substance in the H&E stain slide. So this is very characteristic of the trachea. Here you can see the various cell types. The predominant cell type is the ciliated columnar cell. You can see here, this is the ciliated columnar cell, and this is the structure of the goblet cell. The goblet cell has mucinogen granules in the apical part. These mucinogen granules are washed away during preparation and they appear empty or clear in the H&E stain slide. And the nucleus is present at the base. And the brush cell is cuboidal in shape with microvilli, and the base makes synaptic contact with the nerve terminal, and these cells, they function as the general sensory and chemoreceptor cells. And the granule cells, they have secretory granules, and they secrete the hormones which are responsible for various pulmonary reflexes. These cells belong to the enteroendocrine group of cells which are found in the walls of GIT. This Clara cell or the club cell, it is also a cell type of the respiratory epithelium, but it is seen in the bronchiolar level. So we are going to discuss this cell when we are seeing the structure of a bronchiole.